Hello, this is an extra video that I've made for you to talk about the context of the ELISA that we did and also about other antibody uses. So our scenario was that you're a fruit farmer and you have crop inspectors visiting. They find the mouldy fruit. It could be botrytis, could be something else. These inspectors say that if it is botrytis, they will cancel the £100,000 order, but they'll give you 24 hours to prove that it's not botrytis, if you can then explain how you did that. Your bank manager isn't sympathetic and your insurance won't cover you, so you've got to investigate yourself. We came up with the idea of using proteins to identify the botrytis, and that means we're talking about antibodies. An anti-botrytis antibody is specific and recognizes the shape of an antigen on botrytis. Using antibodies in this way is called immunoassay. So this is how it works. We add samples from our raspberries to wells. This is a fresh raspberry, healthy raspberry, and this is an infected raspberry. The proteins and other molecules in the raspberry mush will bind to the plastic of the little micro well. If we wash everything, if we allow it to bind and then wash everything else away, we're left with this, and we wash with a detergent. The detergent sticks to the sides of the plastic so that no other proteins can now bind unless they bind directly to the proteins that are already present. So we add an anti-botrytis antibody that was made in a mouse. The antibody will bind to botrytis antigen if there's any present, as shown on this side. And if we wash the remaining antibody away, we are left with this. We then have a second antibody that recognizes mouse antibody. So this is goat anti-mouse antibody. So the goat anti-mouse antibody will bind to mouse antibody if it finds it. Again, on the right hand side there is some. And that binds and we wash everything else away. This second antibody is conjugated to an enzyme. The enzyme binds a substrate and catalyzes a reaction that creates a blue color, and the well will eventually go blue. And the more botrytis antigen present, the bluer the liquid will go. The more substrate is converted to blue coloration. So that's how that antibody works. Now, very briefly, I'll mention a different type of ELISA. It's called a sandwich ELISA. In that case, the first step is represented here, in that you add an antibody first to the plastic well and then seal the well with detergent. So you've then got antibodies coating the bottom of the well. You then add your sample, your unknown sample, and if antigen is present, it binds to the antibodies as shown on the left, and then we wash everything else away as shown on the right. We then add a second antibody, which is the conjugated antibody, but this time this antibody is also an antibody directed at the same antigen. It just binds to a different part of the same antigen, and so you get this sandwich effect here. The more antigen there is, the more blue coloration you get as before. And finally, I want to talk about two things. There are antibodies that fight cancer. So you can take an antibody that recognizes a tumor antigen, a protein that is only produced in a cancer cell. Those antibodies can be linked to drugs that kill cells. So if the antibodies bind to a cell that has cancer, it's cancerous, then the drugs will kill that cell. Now, as we've already discussed, if we put a mouse antibody into a goat will produce anti-mouse antibody antibodies. So if you put a mouse antibody into a human, even if it's to fight cancer, the human body will react against it and produce antibodies against that antibody. So what you can do is you can humanize antibodies. You can change the antibody in the areas where it is different from human antibodies by changing specific amino acids. And you do that in the constant region. It's the constant region of the antibody that we're worried about. So you can mutate the DNA of the clone and make specific adjustments to humanize antibodies so that there isn't an immune response to the antibodies when they're injected into humans. And finally, comparing monoclonal and polyclonal. Polyclonal antibodies are antibodies from more than one B cell, so they would recognize different antigens on a pathogen. Sometimes that's an advantage to have 5 or 6 different antigens that are recognized on a pathogen might make the response more specific. It's also easier to use polyclonals and prepare polyclonals. Monoclonals are when one single B cell has been isolated and fused with an immortal cell to produce a hybridoma, and so it's fused with a cancer cell, which is an immortal cell, to produce a hybridoma, and that hybridoma produces one type of antibody only, very specific, recognizes one antigen. Sometimes that's preferable, but polyclonal antibodies can also sometimes be particularly useful.